students, welcome back to my biology online class. In my previous class I have discussed with you about G protein coupled receptor. Today I am going to discuss with you about a very important and a very related topic with G protein, very important topic. That one is second messenger. What is second messenger? All of we know about ligand. What is ligand? All of we know that ligand is secreted by an excited cell. Ligand is bind with a cell surface receptor, G protein coupled receptor. In my previous class I had already discussed with you, and this ligand activates the G protein coupled receptor along with the G protein, and now G protein is activated. Then what happens? The next process will going to start from this point. The activation of an individual G protein may cause activation or inhibition of a particular effector. G protein is now activated. It causes either activation or inhibition. Both of case can happen. Activation or inhibition of effector protein. What is this effector protein? Who will going to affect some, who will going to perform some function, either activation, either it will going to act as activator or it will going to act as inhibitor. Anyone, both of this can happen. The major effector proteins of GPCR are, concentrate this very carefully, these are very much important. First one, first effector protein, adenylate cyclases, phospholipase, C beta. Next, GEF of monomeric G protein rho. Next, beta adrenergic receptor kinase, GI/0 regulated potassium channel, voltage controlled calcium channel, PI3 kinase type gamma. All of these are effector proteins of G protein. Now come to the next step and another conception, GS. What is GS? Stimulatory G protein, which is known as GS. S for stimulatory. When G protein is involved in enzyme activation, it's now known as stimulatory G protein, which is known as GS. And in case of GS, the alpha subunit means GS alpha. The alpha subunit GS alpha activates its effector adenylate cyclase is an effector. Next, this GS alpha subunit binds and hydrolyzes GTP. It binds and hydrolyzes GTP, activates plasma membrane bound this adenylate cyclase. This adenylate cyclase is bound with plasma membrane. And what's about adenylate cyclase? This adenylate cyclase is a large multipass protein and it is transmembrane. Large multipass transmembrane protein. Adenylate cyclase is a large multipass transmembrane protein. Transmembrane means the protein transmits, it crosses the membrane. Some part is outside the membrane and some part is inside the membrane, and that's called transmembrane protein, and its catalytic domain means it surely will have two domain. So its catalytic domain is on the cytosolic site of the plasma membrane, and this one is activation sign, and this activated adenylate cyclase synthesizes cAMP ATP, and this CAMP is a very much important secondary messenger. And from this point, this concept secondary messenger, today our main target, main lecture is about secondary messenger, and this secondary messenger is activated by after activation of G protein, and one of its effector protein is adenylate cyclase, and from this adenylate cyclase the synthesization of cAMP from ATP occurs through these steps. Okay, and each and every step of this, from here to here, simulation of stimulatory G protein and this bind, all of these steps are very much important. Then come to our point of interest, how cAMP is being activated, and what is the present scenario of cAMP when all of this occurs, means what is the first step. Now what's the scenario now? CAMP is being activated from adenylate cyclase, and the CAMP, the first step of activated CAMP is it activate CAMP dependent protein kinase A, pKa. What happens? cAMP now activate CAMP dependent protein kinase A, pKa. cAMP, CAMP also induces opening of certain type of ion channels in the plasma membrane of some highly specialized cells. Protein kinase A, it catalyzes the transfer of terminal phosphate group from ATP. All of we know that ATP has 3 phosphate, P, and terminal phosphate transfer. This one is terminal phosphate group. So transfer of this phosphate group done by protein kinase A. It catalyzes the transfer of the terminal phosphate group of ATP. It will go to specific serine or threonine. All of we know this serine and threonine, these are name of that 21 essential amino acids. Can you remember about serine and threonine? There are 21 essential amino acids. Amino acid means there must be some protein structure. Okay, so specific these ATP terminal phosphate group was transferred to serine threonine of selected target proteins, and after that their regulation, thereby regulating their activity, regulation of activity. Okay, now let's concentrate about some diagram. This is the work of cAMP. Okay, after activation, here what this cAMP activates cAMP dependent protein kinase A, and this activated protein kinase A, it catalyzes transfer of terminal phosphate group from ATP to specific serine threonine of target protein, and this phosphate and the energy released by this action, it will going to regulate the activity of this target protein. Okay, now see, prior all of this happens, what was the condition of normal? What was the normal condition of cAMP and protein kinase A? R, C. R for regulatory subunit, C for catalytic subunit of what? This is cAMP dependent protein kinase A. R for regulatory subunit mentioned here, I am not going to write, and C for catalytic subunit of this cAMP dependent protein kinase A. This is its inactive state also. Now it's activated by cAMP. Here cAMP is coming. R regulatory subunit, C, they are being separated after attached with cAMP, activated, and this was inactive state and this is now active state. This is activated catalytic subunit. Activated catalytic subunit of protein kinase A. Write all of these things. Okay. Then it go to cytosol, and after that it will go to inside the nucleus. This is nucleus and this is all about cell. Okay, imagine this is cell and this is nucleus. So this activated catalytic subunit will go to enter into nucleus. Inactive CREB. What is CREB? I am going to discuss. Inactive CREB is now being active CREB. What is CREB? It stimulates gene expression. Now, what is CREB? The full form of CREB is CAMP response element binding protein. CAMP response element binding protein. So this one is one of the major nuclear target for protein kinase, and this one is activated by CAMP, but this one is CREB is activated by the process of phosphorylation, occurs at a single serine residues, greatly increases the activity of CREB, and this CREB is bound to CRE. CREB is bound to CRE, and what is the full form of CRE? CAMP response element, and this CAMP response element is found in genes. Okay, and this gene transcription is induced by cAMP. So this is the whole scenario about activation of second messenger cAMP and protein kinase A. So this is in a nutshell activation description and working principle of a major secondary messenger cAMP and protein kinase A